Good morning and a warm welcome to all of you to yet another Baltic Breakfast. Today we're going to talk about environmental monitoring. Why is this so important? How come this is the tool to actually help us to do good decisions, to know how to reduce risk, and to put cost-effective actions to preserve our marine areas and our inland waters? My name is Gun Rudqvist and I work at the Stockholm University Baltic Sea Center, and we are organizing these Baltic breakfasts once a month as a way of trying to fulfill our mission to work with outreach. Of course, the Baltic Sea Center is a research unit, foremost a research unit, but we also have a task to try to bridge the gap between research and the rest of society. So the Baltic Breakfast is one of these things that we do sort of regularly. We have, as always, two excellent speakers with us, and we will try to give a lot of time for questions. And you can either post your questions here. You are sitting in the room. It's nice to see so many people here. And you who are online, there will be a Slido code. I think you can see it in the picture, 22102. Or you can also send us an email, ostersjocentrum.su.se. And hopefully there are lots of questions, and if we don't manage to deal with all the questions here right now, we will gather answers from our researchers and put them on the web. So let's move on to today's topic. I mean, there's all of you here, we know that our oceans are really affected by all kinds of human activities, sometimes in dramatic ways and sometimes more subtle ways, of course. And it is key for us to know what is happening and keep long time series, etc., to understand what to do at which moment and with what effort, and how can this be cost effective. I mean, it is so key to have sufficient knowledge. So this is also something that policymakers always ask. What shall we do? What is the most cost-effective thing to do? And without environmental monitoring, it's very difficult to answer these questions. And it's also the fact that there are so many legal, new legal acts or old legal acts that are requiring us to check what's happening. And with different new directives or regulations or national legislation, the demand for environmental monitoring is constantly increasing. But the funds are not following along. So we have a problem, of course, with cutbacks. So today's presentation, we'll be first having sort of a bit of an overview and then diving into one example of a new methods and new ways of trying to make environmental monitoring more efficient. And then we will step on and dive into the chemical aspects and looking at another new way of trying to understand more about all the chemicals in our environment. And with us we have Francisco Nascimento. You are Associate Professor at the Department of Ecology, Environment and Plant Science at Stockholm University. And then the next speaker is Maj-Britt Rian. You are a PhD student at the Department of Environmental Sciences, also Stockholm University. But Francisco, the floor is yours.
Yes, thank you, Gun. Hello, everyone. Good morning. And today, as Gun has mentioned, I'll be talking to you about environmental monitoring and a little bit how we can leverage new technologies to help environmental monitoring deliver on all of these things that Gun has mentioned. But I think we can start from the beginning, perhaps to answer the question why monitoring is important and why do we do it. And I think most of you know this, but we know it's important. It's our best tool to assess large-scale impacts of human activities like eutrophication or environmental toxins on biodiversity and ecosystem health. And it's also the best way we have to follow and track ecological status of marine environments. And also it's an important tool to follow the results and the development of interventions that we find necessary to make in the marine environment. And most of all, it's the tool that we have to then meet the demands of national environmental goals and international European directives and international cooperation agreements. Yes, so what we monitor is then very much connected to this last point. So it is the type of communities and the type of monitoring we do, both chemical and biological, is then to a large extent decided by these environmental goals, these legislations and European directives. So examples of such policies are the Swedish national environmental goals, the Water Framework Directive at the European level, and then the Convention for Biological Diversity, then more at the global level. So these are some of the examples of the types of monitoring we do, and since I'm biased, I chose mostly what we do at Stockholm University. But this is by no means an extensive list. But I think this is an example. The first two, the open water column and the soft bentham, are some of the longest running monitoring programs in Sweden. They started in the 70s around the Asker area and then expanded to larger areas. Since then, like a lot of other types of monitoring have been started, and I want you then to perhaps make a small note of this last two, the soft bottoms and vegetation covered bottoms. That we will get back to them a little bit later. So all of this work is then vital to follow all these points that I mentioned before, but it's also critical for research, for us researchers. So this environmental monitoring is perhaps the best tool we have to follow ecosystem scale responses to environmental change. So one important aspect then of environmental monitoring is then the preservation, the integrity, the preservation of the integrity of this long-term data series, which will allow us then to look at long-term responses to environmental perturbations. At the same time, we have these increasing demands from all these different types of goals, environmental goals, and legislations and directives that Gun has mentioned before, and so there are new demands and increasing demands, and at the same time, as already been mentioned, also we can say that its funding is not stable and predictable, which make all of these things a challenge to meet. Yes, so then let's go back to these two environmental monitoring programs that I ask you to keep an eye on, and let's zoom in a little bit more. So the soft bottom ones is perhaps, I'm a benthic ecologist, so this is the one closest to my heart, and so we can ask the question if the traditional methods we have are sufficient then to comply with all these new demands, to deliver on these. And so how can we then keep integrity of long-term series? So we need to keep doing things as we have been doing, but how do we reply to the new demands? And I think it's not an easy question to target, and I think we can zoom in a little bit into this program, the soft bottom fauna. So for soft sediments, we primarily monitor macrofauna, which are invertebrates, are larger than 1 millimeter, so things that we can see. And as perhaps some of you know, in the Baltic Sea, we have a relatively small, relatively low species richness of these macrofauna organisms. And this is macrofauna then is like the only core indicator that we have for the Baltic, for HELCOM core indicator for biodiversity. So we are then monitoring a low species richness community to comply with indicator of biodiversity. But this low natural biodiversity in the Baltic Sea also creates problems when we want to evaluate the ecological health of the Baltic. We know that that then, because of this naturally low species richness, there's a correlation between the tools we use to classify habitat status, for example, the BQI, with salinity, which results in that some of the areas up north in the Baltic proper cannot achieve like a good quality status because of their naturally low species richness. Yeah, but then the question is, are Baltic sediments then really species poor? Then if we try to squint and look a little bit closer, that's not really the case. So there's a really rich and species-rich, diverse community of smaller animals, what we call meiofauna. There are invertebrates smaller than 1 millimeter and larger than 40 microns. So we cannot see them with the naked eye, but if you take like a fistful of sediment, you can find dozens of species in a normal Baltic sediment. That's one of the groups that we have in meiofauna, the nematodes. We think there are around 200 species in the Baltic. So if Baltic then benthic animals are not species poor, so why don't we look at this? Well, the answer is that it's really hard. So these are very small animals, and you need a lot of taxonomic expertise to investigate these communities and get to the species level. So it's not possible to do it at the scale that is required for environmental monitoring. So the results of this is that we actually doing core indicators for biodiversity while ignoring the most diverse communities that we have in benthic sediments. And this is not mentioning like bacteria and protists communities, like even smaller organisms that have also a high potential to work as ecological indicators. Yeah, so this is where eDNA then offers a huge opportunity then to contribute to ecological and environmental monitoring. So it's a cost-effective and non-invasive technique to detect species using the genetic material that is left in the sediment. So the beauty of the DNA is that you can capture almost simultaneously then several communities at the same time. You could target these beautiful nematodes, or you can also do bacteria and protists if you desire. So I think for benthic monitoring, eDNA shows great promise and can help us then to deliver on these demands of environmental monitoring in a more cost-effective way. But the question is then, how do we scale up these? Then if you remember the second one, the vegetation-covered bottoms, it's another important monitoring program. And these rely heavily then on diving to assess the species composition and the depth distribution of macroalgae. So this is you need highly skilled personnel that can dive and taxonomically identify species under the water. And of course, then this limits to a large extent the spatial coverage that you can perform with these types of surveys. Yeah. So this brings us then to the potential of remote sensing. So using satellite imagery is possible then to cover much larger areas than what divers can do. And new tools or tools that are existing and are being developed, like the Sentinel-2 remote sensing, they can help us then perhaps try large-scale monitoring of microphyte covered bottoms with a much larger spatial scale coverage and scale. Yes, but is this all science fiction, or is it actually possible to do it? So I think, in my opinion, there is a lot of potential to make this work. So we'll start with the eDNA, and I want to show you very briefly, some results of a project that we did called nematode barcoding that was financed by Naturvårdsverket and Vatten- och havsmyndigheter. And here it's basically results from a project where we sampled along the Baltic coastline 80 stations at the same time, from the same place, or from the same stations that the normal monitoring program for macrofauna does. And I think what we can see in the graph in the middle is that we can get data on spatial and temporal variation of nematode species richness. That's the green line, compared to macrofauna, which is then the red line. So you see, it's quite clearly, and I think this is only at the genus level, that we have a much larger species richness of these, just this group of meofauna than macrofauna. And we didn't stop there, then we continued and looked how can we then develop indicators that can help us classify habitat status with this new data based on eDNA, metabarcoding. And this is what we have here. So we have year on the x-axis and the habitat status classification on the y-axis, and the white dots are then the normal monitoring, and the black dots with the new technique. So this then tells us that we can get similar habitat status classification with the new technique. Yes, and in regards to remote sensing, then we also have a new study recently from our Estonian colleagues that has shown that Sentinel data in an Estonian bay was with Sentinel data in remote sensing, we were capable to detect the growth of macrophytes in a specific area. So these two lines, these two pictures are then the coverage in the spring, which varies, like warmer colors are a lot of coverage, colder colors are small coverage. From the spring to the summer, and we see we can detect the growth of macrophytes with satellite in these areas. So I think this shows that there is potential for these two techniques, but big questions remain. So can we then apply this metabarcoding to other types of animals and bacteria? How does the sensitivity and accuracy of remote sensing compare when you calibrate it with diving data? And then can we put all of this then into new indicators of environmental change that can be put into existing tools that are in use today? Yes, and to address these challenges, then I want to talk to you a little bit about this new project funded by Biodiversa and in Sweden, the Swedish National Space Agency. So it's called DNA Sense, and this is where we started this year to try and answer these questions and challenges that remain to try and develop eDNA and remote sensing for environmental monitoring. And this is what we want to do. We want to investigate spatial temporal responses of microbial communities that are not today monitoring, and increase the spatial resolution of macrophytes monitoring with remote sensing, and then leverage these advancements to develop biodiversity indicators that can be used then with existing tools, for example, with HELCOM. So this is our vision then to, with DNA Senses, then to improve and leverage these new technologies to ensure that monitoring programs can meet this current and future demands. So by developing these tools that managers can use, these new indicators, we hope we can help Sweden and other EU members to meet their national and international environmental goals.
Thank you, Francisco. So what's the time perspective of this? Are these new technologies far away or sort of around the corner?
Yeah, so there's a lot of work to be done, of course. I think it's hard to answer, but this new project that we have has a 3-year span. And what we're going to try, we're going to only use environmental monitoring data that exists today. Then we're going to use them to develop these indicators, and then calibrate them. Okay, are they good? A good way to test how.
So in other words, you really need the existing data, so it's
Absolutely.
Building on the older stuff.
Absolutely.
So let's not stop with the older stuff, right?
Absolutely. So the remote sensing is dependent then of this calibration that we have, that is against real data that we see, that divers see with their own eyes.
Okay,
Yeah.
so let's keep funding for that as well,
Absolutely,
right?
Yes.
Okay. Thank you very much.
Yeah,
We'll move on. All right. Right, so now another topic and another aspect of monitoring. We'll dive into the different difficult aspects of chemicals. I mean, you all know that there are so many chemicals out there and we know so very little about them. So how can the new technologies help us with this? Maj-Britt, varsågod.
Thank you. Can you all hear me? Yeah. I hope you're all enjoying your breakfast. I will now then shift the subject over to monitoring of chemical pollution. More specifically, I will focus on discovery-based methods for analysis of organic micropollutants in waters. And I will then introduce to you why we should apply these non-target workflows, which is what I'm working with, and show some examples from how we apply this in our lab. Can you hear me better now? Yeah. Okay, so first some background. In EU, we have an ambition for zero pollution, where we aim for a toxic-free environment. And a focus then in the action plan to reach this ambition is to improve, implement better monitoring of chemical pollution in our environmental samples. And with this rapidly increasing rate of production and releases of chemicals, we are not able to keep up with the monitoring and safety assessments with the current methods that we apply. With these traditional monitoring approaches, where we monitor chemicals in the environment or in our waters, we usually apply what we call a targeted analysis. So here we target a small group of chemicals that we know already exist in our samples. So we can quantify these, and then we can do risk assessments and compare with toxicity values. But these methods have never been proactive. They have never prevented widespread environmental contamination, and we have multiple examples of this in the past, where we have discovered chemicals already when it's too late, after we already have a widespread environmental contamination. Just to show one example, you might be familiar with these polychlorinated biphenyl compounds, PCBs. This is a newspaper from the US in 1976, I think, saying that PCB is everywhere. And these PCBs, they were first discovered in the environment by a Swedish chemist called Søren Jensen. He discovered these in 1966, over 30 years after we started using them in the industry, after they already were distributed globally. Still, we are recommended to limit our consumption of fatty fish from the Baltic Sea, because we still have so high levels of these chemicals in our environment. Already back then, Søren Jensen already pointed out that we need methods that can discover pollution in the environment. So we need to complement these targeted methods that we are applying for chemical monitoring with more proactive methods that can accelerate discovery of legacy pollution and also identify new or emerging hazards before they get widespread in the environment. Just to put some number on it, over 350,000 chemicals are registered in global commerce. The majority of these chemicals are not monitored in the environment, and we also don't have information about, for example, their toxicities. We don't know if they're toxic to environmental or human health. And we also lack other information. For example, we don't know if they're persistent in the environment or if they will easily break down. If they break down, what will the product then be? And also, we don't know their mobilities, how they move around in the environment. So out of these hundreds of thousands of chemicals, only a few hundred are routinely monitored. and roughly around 5% have been analyzed in our environmental media. So you can see here that there is a huge knowledge gap, and this knowledge gap we have to fill by applying more discovery-based methods. So that's where this non-target analysis come in, which I'm going to focus on in my talk. So these are methods based on high-resolution mass spectrometry. And what we do here is that we collect all the chemical signals that we can detect in the samples. So we do not only look for the chemicals we already know exist in our sample, but we also look for the unknown chemicals using these approaches. So with these methods, instead of screening for, let's say, 10 target chemicals, we can detect over 1,000 or thousands of chemical signals in one sample. So now I will show you an example how we are working with this. So we have applied non-target methods to look for persistent chemicals in Swedish waters. So we have analyzed over almost 400 water samples, including groundwater, surface water, drinking water, stormwater, and wastewater from all over Sweden. Since we're at the Baltic Breakfast seminar, you might think, why no Baltic Sea samples? But I will just let you know it's relevant because these water will influence the Baltic Sea. For example, wastewater is one of the major sources of pollution in the Baltic Sea. So what we have done is that we have analyzed these samples with our non-target workflow with liquid chromatography, high-resolution mass spectrometry, collecting all the chemical signals that we can detect. And then we pair this up with advanced computational tools that can help us characterize the molecular composition of the samples. And we are focusing on persistent chemicals, and I will show you also how we prioritize these in our non-target data set. So first, I need to show you how our data output look like. So for each of these chemical signals that we detect, we call them one chemical feature. We will have the information about their masses, their exact mass, their retention time from the chromatographic system, and also their intensities in each and single sample. And additionally, we fragment this molecule and collect what we call the MS2 fragmentation spectra. And this is information that is highly valuable to us because it can tell us something about the chemical structure. For example, we can run this experimental spectra, illustrated here in green, through big databases of MS2 spectra to see if we have a match. It's like a fingerprint matching. And if we do, then we can assign a probable chemical structure. Or we can take this one step further, apply computational tools in silico models that can help us predict the chemical structure. So this is how the output can look like when you analyze almost 400 samples. So each of these points here is one chemical feature with its own mass, retention time, and also its own fragmentation spectra. So you can see that we can collect very complex data when we apply these methods. And then we have to prioritize somehow what are we interested in this huge data set, because we can't identify everything here. So depending on your research question, you can apply different prioritization strategies to kind of filter out those features that are relevant for your research question. For example, we were asking, can we detect any persistent chemicals in our water samples? So we checked if our non-target features were persistent through wastewater treatment. And then we prioritized the features that we saw were persistent through wastewater treatment, because then we they might also be persistent in the environment. So we detected over 70,000 chemical signals in our wastewater samples, and over 15,000 of these persisted through wastewater treatment, meaning that these can reach the environment and also the Baltic Sea. So then we checked our other water samples, for example, our groundwater or drinking water. Can we find any of these persistent chemicals in our samples? And then we checked if we could assign some probable structures to these. And I will show some examples from our spectral library matching here. First example, we had a match with this chemical, perfluorobutane sulfonate, which is a PFAS, classic persistent chemical. It was persistent through wastewater treatment, have been previously classified as very persistent, very mobile. And we could detect it with high detection frequencies in our other water samples. For example, in 35% of our groundwater samples and almost 80% of our tap water samples. Another example is this urban biocide here called terbutrin, applied in construction materials and paint and so on. We also classified this one as persistent through wastewater treatment and also detected it in our other water samples. And for this one, we don't have any persistency data available in the REACH database, which was the case for actually most of these persistent features that we have assigned a chemical structure. But other studies have reported that it's insufficiently removed through wastewater treatment, also supporting our hypothesis that this is a persistent chemical. I think I'm running out of time. I wanted to show some more examples, but you can see just very briefly that we have detected a lot of different type of chemicals that were persistent through wastewater treatment and detected in our other environmental water samples. So, just to conclude, I would like to mention some of the benefits with these non-target analysis approaches. First off, they are more discovery-based. We can accelerate discovery of contamination that already exists in the environment if we apply these methods. We can apply it proactively to monitor the environment of new or emerging hazards. We can, for example, use it to answer other questions. Do we have a lot of chemicals with specific properties? We can also use it, we can also, for example, predict toxicity based on the data we have of the unknown chemicals. And we have a lot of different applications for these methods. But we need more research to develop the tools that we need to have efficient workflows, to improve the quality assurance, to increase the number of chemicals that we can identify in the samples, and also to improve the confidence of the results. Yeah.
Thank you. So I'll ask the same question like I asked Francisco. What about time perspective? Is this also sort of evolving rapidly, or are you still at an early research stage?
This have been applied in multiple labs over a long time in research context. And it have already proven that it can be used to discover new chemical pollution. And we have also shown that we can use it to, for example, detect spills of chemicals. And there are a lot of good workflows developed still, but it's not applied in like regulatory monitoring yet.
Okay. So I was thinking about the Urban Wastewater Directive, which recently was decided and now will be in the implementation phase on national basis. Do you think that these methods can play a crucial role here, trying to know what to do with this?
I think so. Also, one of the points in this new directive is to improve the monitoring of chemical pollution, because we are only studying a low number of chemicals that we already know quite a lot about, established pollutants. So I think it can be applied to, for example, monitor the wastewater effluent after it have been treated to see what we are releasing. For example, we can apply it to prioritize which chemicals we should add to the monitoring programs for to do more quantifications and so on.
Okay, thank you. Thank you.
Thanks.
So, Francisco, come and join us here. And we will open for questions, of course. Anybody directly? Or do you want me to start? Yeah, here we go. Ellen, my colleague, will be running with the microphone. Please,
Yeah,
please
so-
introduce yourself. As
Okay, Jesper Ström, and I'm working with the environmental monitoring program, specifically the vegetated bottoms. So we're out diving. Therefore, I have a question for Francisco. And it's the new methods of monitoring with help of satellites and stuff. Can you actually distinguish different types of macrophytes, like brown algae, red algae, with the spectral information that you get?
Yeah, thank you, Jesper, for the question. I'm not an expert in remote sensing, but I think that is like possibly the ceiling of the capabilities we have right now. Is maximum we can get to if it is a brown algae or a green algae. And then there's a limitation also on how deep you can go. Especially in like color complex waters like the Baltic Sea, where the signal stops penetrating earlier than, for example, in the Mediterranean. So there are a lot of challenges that we need to solve first. But as I mentioned to Gun, so this will never be like a substitute for what you actually do, but more a complement, so that perhaps we can increase the scale of what is being done right now.
Okay, thank you. More questions? Yes, we have one here as well. Okay, sorry, we'll start there and then.
Yes, I'm Mikael Sjöwall from the magazine Finland's Natur. I would like to know if there's any plans on boundary crossing cooperation as regards remote sensing with, for instance, Finnish companies or authorities.
Yes. So this DNA Sense project, it's a European project, and Finland is an active partner on that, on both sides. So both on the eDNA and on the remote sensing part. So it's Sike. So we have a team of 4 researchers from Sike that are involved in this. So both in the remote sensing part, but also in this indicator development part.
Maybe I can ask that for your perspective as well. What about cooperation between countries? Is that also part of your project?
In my research specifically, I'm studying only water samples in Swedish, in Sweden. But of course, the methods that we are applying, we are discussing with labs from other countries also.
I had one question here, right? Yeah.
Hi, thanks for your talk. It was fascinating, both of them. I'm Ria Dunkley. I'm from the University of Glasgow, and I was interested to know if you combine the methods that you use in kind of remote sensing and monitoring with community science or citizen science approaches, or is that something that you would consider maybe if you haven't so far?
Interesting, participatory approaches.
Do you want to go?
You go. It's a big question.
I think for remote sensing, it's probably limited possibilities, maybe with the calibration. But for eDNA, there's certainly a potential for citizen science. We're not using it right now, but in this project. But I know there are within the same call that we got funded, there are several projects that use citizen science for eDNA projects. So to help increase the number of samples that are being collected.
Sorry.
I'm not sure if this is really answering what you were wondering, but you can also, like for the chemical pollution, I mean, you can also see, for example, which type of medication is used within the society, and then use this as metadata to help us identify chemicals with higher confidence.
Okay. Thank you.
Yes, my name is Per Bolland. I'm a researcher at Stockholm Resilience Center. So I have two questions. The first one is on measuring in the source or upstream, or measuring in the actual environment. Isn't there a risk if you only measure upstream that you miss other sources? For example, if you measure in the Baltic Sea, you don't get only Swedish emissions, you get emissions from other countries, which you would miss if you only measured in Sweden, so to speak. So that's the first question. The other question is on monitoring more generally. Is your view that Sweden is fulfilling our commitments to international commitments that we've done when it comes to monitoring, or do we have to increase monitoring? And would you say that we have to increase it, in that case, a lot, or are we close to where we should be? Okay. Thank you.
Thank you. I think partly you did answer that, Francisco. You know, we're not doing enough, right, on monitoring in general.
I think what we need to be mindful of is to not miss opportunities, right, to capitalize on new technologies and how they can help making the existing monitoring more cost-effective and answer the questions in a better way that we're answering now. I mean, I'm mindful that as a society we have limited resources, but I think there is space and a need to increase funding by a certain amount, but also to be mindful and aware of where we can improve things.
Yeah, I want to answer the first question you had about the sources of the pollutants. For example, if you study only the wastewater effluents from one specific city, we will not get the full picture, of course. But you can also apply the new methods to actually, if you do strategic sampling, you can use these methods to also track where are the pollution coming from.
Can I just add something more? I think there is a cost also of not identifying correctly the pressures and drivers that are impacting our environment. And if we're doing that in an insufficient way, there will be a cost later on for society. So we need to keep that in mind also.
Yeah, that's good.
Yes, hello, my name is Björn Brindefalk. As someone else also working with eDNA, I have a question for you, Francisco. So I saw that you had chosen an amplicon-based approach for this, and I understand that this is at least partly an economical question. But what is your take on the choice between amplicon-based methods and just going full shotgun, since that opens quite a lot of other possibilities?
Yes, that's a good question. So we actually tested that for the communities, or these roundworms communities, nematode communities that we have investigated. So with mock communities, we tried both amplicon-based techniques and shotgun sequencing, and they performed. I would say metabarcoding performed better even than shotgun, which was for us surprising, because shotgun doesn't have all these PCR biases that we put. So we, in this DNA Sense project, we have also metagenomic data specifically for communities, for bacterial communities. But I think for animals, because the signal is much weaker in eDNA, I think metabarcoding is a good choice.
Okay. So a final question I think we have here. Yes.
Oh, okay, can I take it? Yes. I was just wondering, would it be possible to actually go for the 73,000 features that you had and identify them if you had enough data power and enough time and money?
I mean, I will not say that nothing is impossible, but it would demand a lot of effort to actually be able. It takes a lot of time to do these identifications, and also a lot of these are compounds that are naturally occurring. It's background from the sample. A lot of natural occurring compounds. Yeah, I think you would need a lot of staff to be able to do that kind of work.
And also, I suppose, I mean, the chemical industry is putting out releasing new chemicals in a quite fast pace, I suppose, yeah.
Yeah, so fast that we struggle to follow.
Of course,
keep
yes.
up with the monitoring.
And then we come to the matter of availability of data. Is that a
Yes.
problem, I suppose?
That's one of the things that are so great with this non-target data set is that we haven't target specific chemicals and we collect so much data. So if we share this data, you can, another institute can go in, check my data. Do we find this chemical that we are interested in? Or yeah, if we do this over time, we can see when some new chemical signals pop up, and then we can focus our attention on these new introduced pollutants.
As always, time is running out. I just want to ask you, finally, you are now the policymakers. You can make all the decisions. What do you want to see tomorrow being done?
Should I start?
Yeah,
I will
go
answer as an
ahead.
analytical chemist then. I think we should put more money into regulating and monitoring the pollution so we can be more proactive. We would save money if we detect the problem early so we don't have to actually clean out everything after the pollution is already there. Yeah.
Francisco.
I think, well, I agree with the more money part, of course. But I think there's also potential to increase international cooperation, for example, within the Baltic, so that we can standardize and share data in a much more efficient way than we do. Standardize methods and share data in a more efficient way than we do today, and actually to follow where, for example, chemicals, the origin of the chemicals, so we can prioritize actions. So what are the most effective actions and where they can be taken, than everyone doing their own thing separately.
Thank you ever so much. A warm applaud, I think. And thank you all for attending, all on the line and all here in the room. I hope you have a really nice day. You're welcome back in November, and then we will talk about plastics. So check an eye on our web or our newsletters so you can get the right date and the information. Have a nice day.